Hi everyone. In this lecture, we are going to focus on eukaryotic cells. In the last lecture, I said that we were talking about prokaryotic and eukaryotic cells, and the other lecture focused on prokaryotic cells. So this lecture is going to focus on eukaryotic cells, and because of the nature of the microbiology class, we're mostly focusing on bacteria in class. So bacteria, again, are prokaryotic cells, but humans and animals and everything else that's not archaea or bacteria are eukaryotic cells, and it's important to understand the structure of eukaryotic cells. So for example, when you design a drug that's supposed to target bacteria, it targets the bacteria and not the human eukaryotic cells. So eukaryotic cells can be humans, animals, plants, protists, mold, fungus, yeast, all of these things. In the background of this lecture, we're going to do one last overview of prokaryotic cells versus eukaryotic cells, and then I'm going to briefly talk about cell size in eukaryotic cells. They are bigger than prokaryotic cells, and then we are going to talk about the components that are in eukaryotic cells, mainly the differences that we do not see in prokaryotic cells. So we're going to talk about all of these components listed here. Okay, to start off, again, to summarize, everything that's alive is made out of cells, and there's only two types of cells in the world. You're either made up of prokaryotic cells and eukaryotic cells. Things made up of prokaryotic cells are bacteria and archaea. Everything else that is alive is made up of eukaryotic cells. And again, viruses are not made up of either because viruses are not alive. So prokaryotic cells generally are smaller, simpler. They don't have a nucleus. They don't have membrane-bound organelles. And prokaryotic cells are what make up bacteria and archaea. Eukaryotic cells are larger. They're more complex. We see organelles in them. We're going to talk about these different organelles. Organelle, actually, let me mention it here. So everyone, you've all heard of like organs in humans. Organelles are similar. They're like the organs of cells. Organelle means like the small little components of cells. And then eukaryotic cells make up everything alive that's not bacteria or archaea. So you can see them in plants, fungi such as mold and yeast, and animals. Okay, so we talked about all of these, and then another thing in prokaryotic cells is they have one circular chromosome, and chromosome and DNA are the same thing. Their DNA is concentrated in a region called the nucleoid. Again, the nucleoid is just the region where we see the DNA. It is not the nucleus because prokaryotic cells do not have a nucleus. They do not have membrane-bound organelles or histones, and the cell walls in bacteria, if they do have a cell wall, it's made up of peptidoglycan. In archaea, if there is a cell wall, it's made up of pseudomorine. So different chemicals, different compounds. And then prokaryotic cells divide by binary fission, meaning they just split. When we talk about DNA and genetics later, we'll talk about all the details of binary fission. Eukaryotic cells are found in animals, plants, protists, fungi. They're larger and more complex. They have a nucleus. The purpose of the nucleus is to house the DNA. The chromosomes or DNA are found paired inside the nucleus. There are membrane-bound organelles and histones in eukaryotic cells. If a cell wall is present, such as in plant cells, it's made up of polysaccharide, meaning sugar. But human cells, and I'll talk about this in a little bit, do not have a cell wall. And eukaryotic cells divide by mitosis. Okay, now prokaryotic cells, let's just compare the cell size. Prokaryotic cells are generally much smaller than eukaryotic cells. Bacteria are generally much, much smaller than human cells. So prokaryotic cell size are about 1 micrometer. Micrometer and micron mean the same thing. And to give you an idea of how big that is, here is the tip of a pencil is about 1 millimeter. If you divide that 1,000 times, that size is 1 micrometer. So bacteria are usually an average size of 1 micrometer, meaning if you take this tip and you split it 1,000 times, eukaryotic cell size can range from 10 micrometers to 100 micrometers. Micrometers, again, and microns are the same thing. Okay, now we're going to summarize what do we see in all cells? What are things that are similarities between cells? This is a very good test question that I will probably ask you about. So list the similarities that we see in all cells. What all cells have is all cells have a plasma membrane, which is just the outside of the cell that protects the cell components. All cells have cytoplasm, which is just the inside, everything inside the plasma membrane. All cells have DNA, but remember there is difference between eukaryotic and prokaryotic DNA. Prokaryotic DNA is just one circular chromosome. In eukaryotes, they're paired and linear. But just to make it easy, on a test you will write all cells have DNA, and then finally all cells have ribosomes. But in prokaryotes, it's a 70S ribosome. In eukaryotes, it's an 80S ribosome. So these are the similarities between cells. Okay, now we're going to focus on eukaryotic cell structure. So I'm going to talk in detail about each one, but what we're going to cover today in this lecture is I'm going to cover, again, the structure and function of the plasma or cell membrane, and then what's inside the cytoplasm. So we're going to talk about all the organelles, ribosomes, briefly about the cytoskeleton, and then the cell wall if it is present in eukaryotic cells. And then some eukaryotic cells have flagella and cilia, which we will talk about. Okay, let me skip. So if you are studying this slide, I just wanted to tell you, I was trying to show you the difference between plant cells and animal cells. Both are eukaryotic cells. Plant cells have cell walls, which animal cells do not, and plant cells have an organelle called a chloroplast. That's why they can do photosynthesis. We animal cells do not have neither. We don't have a cell wall. Our cells do not have an outer cell wall, and we don't have chloroplasts. But then inside animal cells, we see in some animal cells flagella, such as in sperm cells. We see a lysosome, which I will talk about later. Okay, so now let's get into the structure of eukaryotic cells. So eukaryotic cells, like prokaryotic cells, have a plasma membrane, a cell membrane, a cytoplasmic membrane. It's all the same thing. And it's very similar structure as in prokaryotic cell membranes. So all cell membranes are made up of a phospholipid bilayer with proteins. And we already discussed the significance of that in the prokaryotic cell structure. The differences that we see in animal plasma membranes that we do not see in prokaryotic cells is that animal eukaryotic cells have sterols in their phospholipid bilayer. Sterols are just fats, they're complex lipids, and an example of a sterol is cholesterol. So we have cholesterol in your phospholipid bilayer, and the reason why we have these components is because they maintain the integrity of the cell membrane. That's just a fancy word of saying that they make it like a better protectant. So they help it be not your cell membrane, not be too fluid, not let things out, because we don't have a cell wall. So it's another layer of protection. Now, sterols are not found in plant cells. You cannot get cholesterol from eating a plant-based diet, just if you're trying to memorize it. So we see sterols in animal cells, but not in plant cells. Plant cells, what they have to maintain the cell membrane's integrity is they have a cell wall and it's made up of polysaccharides. So here is, again, what the plasma or cytoplasmic membrane looks like. It's a phospholipid bilayer, so 2 layers. It's phospholipid, so phosphate heads, lipid tails, and then there's proteins integrated all throughout the cell membrane, and the proteins play a function in transport. So the function of the cell membrane is very similar to prokaryotic cells. It's to protect cells, to help cells sense their environment, and it's semi-permeable, meaning it lets some things in and other things not, some things out and other things not. And probably the most important function besides protection for the plasma membrane is transport, so letting things in and out. And in the plasma membrane of eukaryotic cells, we see both passive and active transport, just like we discussed with prokaryotic cells. Here's a good just review video on this.
The plasma membrane is selectively permeable. It lets some substances through and not others. The hydrophobic interior of the bilayer is one reason membranes are selectively permeable. Hydrophobic molecules, those that are soluble in lipids, can easily pass through the membrane. In addition, small molecules like O2 can sneak between the phospholipids of the membrane. On the other hand, large hydrophilic molecules like glucose and ions, such as sodium ions and hydrogen ions, cannot pass through the membrane unaided.
So for big molecules that need to get across the membrane, both in and out of cells, we have proteins. We have integral cell membrane proteins. That's their function. And so differences that we see in the cell membrane in eukaryotic cells that we do not see in bacteria is eukaryotic animal cells do a process of transport called exocytosis, and I will talk about it. It's a way where the cell membrane basically rearranges to kick something out of the cell. Endocytosis is when the cell takes up something. And these two processes take up a lot of energy, so they're active types of transport. And specific types of endocytosis, when the cell is drinking, we call that pinocytosis. That's a type of endocytosis, taking components inside the cell. And then cell eating is phagocytosis, which is a type of endocytosis. And with phagocytosis, if any of you have taken another like physiology or anatomy class before, the importance of it is in the immune system, which we will cover in much detail later on. Okay, so here's a good video just summarizing transport in plasma membranes.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells?
That's passive transport, remember, is no energy required, and you're going from high to low
Lipids,
concentration.
such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis.
So that was passive transport, diffusion, facilitated diffusion, and osmosis.
The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is co-transport.
Okay, I'm going to stop it here. Co-transport is just when you have two transporters, two protein transporters. But the main take-home message I want you to know is there's passive transport and active transport. And examples of passive transport are diffusion, facilitated diffusion, and osmosis. And then active transport requires energy, and it's when molecules are going against their concentration gradient, meaning they're moving from low to high concentration. So I hope that by now, especially after watching the prokaryotic cell lecture, everyone understands this. So passive, no energy required, substances move from high to low concentration. Simple diffusion is an example. Facilitated diffusion is when you have a protein involved in the cell membrane. And then osmosis is just the movement of water across the cell membrane. Active transport, also important in the plasma membrane, is when substances go from low to high concentration, which is the same thing as saying that things go against the concentration gradient. Examples of that are just active transport, meaning you have energy and you're going against the concentration gradient, or endocytosis and exocytosis. Endocytosis and exocytosis we only see in eukaryotic cells, so that's why we didn't talk about them in prokaryotes, but it's basically endocytosis is when the whole cell membrane arranges to take up something, so that takes energy. That's why it's a type of active transport. And then exocytosis is when the whole cell membrane rearranges to kick a substance out, and it also takes a lot of energy, so it's a type of active transport. And then here I have my important question: which of these are present in bacteria or prokaryotes? Simple diffusion is present, facilitated diffusion is present, osmosis is present, and active transport is present. Endocytosis and exocytosis are not present, they are only in eukaryotic cells. Okay, so again, just summarizing what I said, and then in the prokaryotic cell lecture, we talked about hypotonic, isotonic, and hypertonic solutions. Okay, I'm gonna skip more videos. Okay, so again, osmosis is the diffusion of water across the cell membrane. Water goes both ways, it goes in and out of cells. So it's important for us to know what type of solution we put cells in. Water will always go to where there's less of it. So water will go where there's less water or where there's more salts. Because if there's more salts, there's less water. So this is the same thing as saying that water moves to where there's higher solutes or higher salt concentration. So cells contain mostly water, but there are solutes inside. When you place a cell in a solution, water will always move where there's more salts. So if there's more salts inside the cell, water will move in the cell. If there's more salts outside the cell, water will leave the cells. So types of osmotic solutions that we talked about in the other lecture, a hypotonic solution has less solutes or less salts than the cell. So in a hypotonic solution, because there's less salts outside than inside the cell, water will go where there's more salts inside the cell. So it will keep going in the cells and the cell will eventually swell and burst. In an isotonic solution, the solution has the same amount of salts as the cell, so water enters and leaves the cell at the same rate, so nothing happens to the cells, they're happy and they're healthy. And then if you put a cell in a hypertonic solution is a solution that's hyper, so it has more solutes or salts than the cell. Water will always go where there are more salts, so water will leave the cells and the cells will eventually shrivel. So hypotonic and hypertonic solutions in theory are both bad because you will damage cells. So this is also again demonstrating what I said. Okay, now the reason why this is important with eukaryotic cells, so I talked about it in prokaryotic cells, we need to know how to grow bacteria in labs, so we always want to put them in isotonic solutions. But the reason why this is important to learn with eukaryotic cells is you always want to make sure, I know a lot of you guys want to go to nursing school and healthcare fields, when you give someone an IV is not pure water. An IV has a different concentration of solutes, and the reason is that we want to keep a person's cells healthy. If you were to just give someone just plain water with no salts, that would be considered a hypotonic solution, and that means that water would enter someone's cells and they would burst. So that's why we want to be careful with IVs. So in a hypertonic solution, water leaves the cells, so the cells shrivel. In an isotonic solution, water goes in and out of cells at the same rate, so cells stay healthy. In a hypotonic solution, water enters the cells, so the cells swell and will eventually burst from all the water. So again, this is why it is very important to be careful with the type of IV solution you put. And there are many different concentrations of salts for IV solutions. So it's not, there's just like the traditional one, but sometimes we have patients who have a lot of salts in their blood or they have very little salt. So sometimes we have to adjust the IV depending on the salt concentrations. Just keep that in mind for those of you going in the health field. Okay, so now let's talk about active transport. Active transport, again, is just when you require energy to move components across their concentration gradient. And with active transport, you always also require a membrane protein. So here's a good video demonstrating it.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients.
So that's it. That's all I want you to know, that you require energy. There is a membrane protein and its molecules going against their concentration gradient. And then finally, another example of transport that requires energy that we only see in eukaryotic cells and animal cells is exocytosis and endocytosis. Types of endocytosis are pinocytosis and phagocytosis. Pinocytosis is when the cell rearranges the whole cell membrane to take in a lot of liquids, and phagocytosis is when the cell membrane rearranges itself to take up particular material. So phagocytosis, usually your cells are eating bacteria and trying to digest them so that they're not causing an infection. And then exocytosis is when you kick something out. You rearrange the cell membrane to kick something out. So both require high amounts of energy. So they're both types of active transport that we do not see in prokaryotic cells. So prokaryotic cells do not do endocytosis and exocytosis. And again, two examples of endocytosis are pinocytosis and phagocytosis. Okay, I think I explained this. You guys have this slide now, and here's a summary of all the transport. I hope after going through all this transport in much detail with both lectures that you all know it. I know it comes up on a lot of various exams, like health exams that you transfer to, they ask a lot about this. Okay, now we're gonna move on. So in eukaryotic cells, some cells have a cell wall. Cell walls are not present in human cells. The algae, plants, and fungi, which are all made up of eukaryotic cells, have a cell wall. The cell wall is not made up of peptidoglycan. It's made up of sugars, of carbohydrates, of polysaccharides. So carbohydrates, polysaccharides, and sugars all mean the same thing. And it's there to provide more support for plant cells. If you think about it, plants are exposed to so many things that are out in the environment. So it makes sense that they, outside the cell membrane, they would have another layer of support called the cell wall. And again, animal cells do not have cell walls, but what do they have in their cell membranes for support? I always ask this to students in class, and we have cholesterol or sterols, which we do not see in animal cells. So cholesterols and sterols are a good thing. Just too much and a type of cholesterol is bad, but in general, you see it in our cell membranes. Another component that we see in eukaryotic cells are flagella and cilia. They're present in some eukaryotic cells, not all eukaryotic cells. So both flagella and cilia are just projections outside the cell that are important for cell motility and moving substances along the cell surface. So flagella are long projections, and they're few in number on eukaryotic cells. So an example of where we see flagella on eukaryotic cells are sperm cells have flagella, and the reason is they need to swim to fertilize an egg. Cilia are little short projections here that some cells have. For example, some of your respiratory cells have cilia on them, and they're important to move substances and microbes out. So it's a way to sweep up any bad microbes, bacteria, viruses out of the lungs so they don't get there. So it's a way to protect you. Same thing, trying to kick viruses out, such as coronaviruses. The flagella and cilia, so what I want you guys to know here is that flagella and cilia are different than the flagella found in prokaryotic cells. The flagella found in prokaryotic cells is made up of the flagellin protein. The flagella found in eukaryotic cells is made up of the tubulin protein, and it's powered by ATP. That's all I want you to know here. And then in eukaryotic cells, we also have cytoplasm. Cytoplasm is just a substance inside the cell membrane. And so basically it's everything between the cell membrane and the nucleus. So all the watery substance. The watery substance is called the cytosol and the organelles. And then we also see the cytoskeleton. The cytoskeleton are just various proteins that hold the structure of the cell together, similar to our body structure. And then in eukaryotic cells, you find ribosomes in the cytoplasm, just like prokaryotes, but our ribosome is ADS. Okay, now let's talk about ribosomes. So the function of all ribosomes is to make proteins, and in the prokaryotic cell structure, I told you that prokaryotic cells have a 7DS ribosome. Eukaryotic cells have a completely different ribosome, but it's the same function. All the function of ribosomes is to make protein. I guarantee you 100% there's going to be a test question, which is, what is the function of a ribosome? So I want everyone to know that the function is to make protein. And the structure, ribosomes are made up of proteins and a molecule called ribosomal RNA. The ribosome in eukaryotes is called an 80S ribosome. It consists of a 60S large subunit and a 40S small subunit. Remember, these do not add up to 80 because it has nothing to do with addition. The S stands for sedimentation rate. So when you put it in a machine and lab and you spit out how long it takes to go down. So I want everyone to know the take-home message of this is eukaryotic cells have an 80S ribosome, prokaryotic cells have a 70S ribosome, and this will for sure be a test question. We see ribosomes membrane-bound and free in the cytoplasm. They're everywhere in eukaryotic cells because they're very important, and their function to make protein is very important. And again, the ribosome in prokaryotes is 70S ribosome. Okay, another thing that we see in eukaryotic cells is a nucleus. The purpose of the nucleus is to contain or house the DNA or chromosomes. DNA and chromosomes are the same thing. When we look at the nucleus, there is a nucleolus. The nucleolus is where ribosomes are made in the nucleus. And then the nucleus is surrounded by a nuclear envelope just to protect it even more. And nuclear pores allow materials to get in and out of the nucleus. So for example, you're going to learn later on that there's an important genetic molecule, RNA, that has to get in and out of the nucleus, and we'll learn about it later. The nucleus houses our DNA or our chromosomes. Our DNA, unlike prokaryotic DNA, is wrapped together with histones. They're proteins that wrap up your DNA to keep it protected to form chromatin. When the chromatin condenses, it condenses into chromosomes. That's all I want you to know. Chromatin, chromosomes, and DNA all mean the same thing. They're all different forms of DNA, but they're all DNA. Okay, now to get to the mitochondria. So the mitochondria is an organelle that we see in eukaryotic cells. It's the power plant of the cell, meaning it makes energy, meaning it makes ATP. So the mitochondria is probably the most important organelle found in eukaryotic cells. It's involved in cell respiration. We will learn about metabolism next week. And in cell respiration, that's where we make our energy, which for our cells is ATP. Mitochondria, when we look at them, they have a single circular DNA of their own. They also have 70S ribosomes. We saw 70S ribosomes and circular DNA in prokaryotic cells. So this suggests that mitochondria have a bacterial ancestor or maybe developed from bacteria. So here is the mitochondria. Their function is to make energy, and its structure is very important. The main thing I want you to know is when we look at the structure of organelles, there's a lot of folds called cristae, and these folds are important because this is where cell respiration happens to make ATP. The more folds you have, the more ATP you make. So this is the structure of the organelle. Another membrane-bound organelle that we see in eukaryotic cells are chloroplasts. We only see chloroplasts in plant cells. We do not see them in animal cells. The function of chloroplasts is to do photosynthesis for plants, so they make ATP in plants and algae. And chloroplasts also have a single circular molecule of their own DNA, and they have their own 70S ribosome. So we also think that chloroplasts have a bacterial ancestor. Another membrane-bound organelle is the endoplasmic reticulum. The function of the endoplasmic reticulum is it's the factory of the cell. It's where all the macromolecules are made. So so you have the nucleus. So the nucleus houses the DNA. Outside the nucleus, you have the endoplasmic reticulum. This is where cell components are made. And we have smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth ER is called smooth because it doesn't have anything on it. Rough ER is called rough ER because it has ribosomes on it. So rough ER is where we see proteins made because that's the function of ribosomes. So rough ER has ribosomes, and this is where we receive, transport, store, and make proteins. The smooth ER is where you make everything else. You make the fats and the cell membranes and the hormones. So that's the function of both, but overall, all the ER, smooth and rough, is a factory of the cell. And the protein is made on ribosomes. Ribosomes are on the rough ER. Just keep that in mind. The Golgi complex, the Golgi apparatus is another organelle that we see. It's called the post office of the cell because the Golgi apparatus is a part of the cell that basically, once things are made in the ER, it tells them where to go. It packs and sends end products out of the cell. So it's a series of sacs independent of the endoplasmic reticulum. Transport organelle, it modifies proteins from the ER and it assembles them together and routes them, meaning it tells components where they need to go. So for example, if you had a protein made on the rough ER, and let's say it was a membrane protein, that protein would go to the Golgi apparatus, and the Golgi apparatus would mark it and say, you need to go to the cell membrane because that is where you belong. So that's the Golgi apparatus is the post office. It routes end products by all these vesicles. So if you have a molecule that you need, like a protein that needs to go to the cell membrane, it'll put it in a vesicle or a vacuole and it'll send it to the cell membrane. So vesicle transport, vesicles transport material in and out of cells. So if you have protein made on rough ER, they'll get sent to the Golgi apparatus. The Golgi apparatus will send them to their final destination through a vesicle, and that's what vesicle transport is. Lysosomes, eukaryotic cells have lysosomes. Lysosomes are not found in prokaryotic cells, and they're basically the garbage dump of the cell. So this is where we destroy toxic material, extra material that the cell doesn't need anymore. There are these vesicles inside our cells that have a lot of digestive enzymes, and they're made in the Golgi complex, and their function is to digest old organelles so that we can reuse the material. They destroy harmful bacteria, and when we look at cells that do phagocytosis, such as your immune cells, your white blood cells, we see a lot of lysosomes there. And that reason is they're trying to target harmful bacteria and viruses. So it's good to have all these lysosomes, which are sacs filled with digestive enzymes. So here's an image. So you have a lysosome, here's a damaged mitochondria, and these vesicles will fuse. The digestive enzymes in the lysosome will digest this old organelle or old bacteria, and after it digests it, it lets the material out so we can recycle anything that the cell can use again. And then finally, I want everyone to know something called endosymbiotic theory. Endosymbiotic theory suggests that eukaryotic cells originated from prokaryotes, and the idea is that large bacteria engulfed small bacteria, so they ate each other, and aerobic bacteria became mitochondria, photosynthetic bacteria became chloroplasts. The evidence for this theory is when we look at chloroplasts and mitochondria, both of them have two things: a circular piece of DNA that we see in prokaryotes, and also they have a 7DS ribosome. So that's the evidence for the endosymbiotic theory, saying that our cells generated from other bacteria. So we covered eukaryotic cells, so I want everyone to go through all the components, make sure that you know where we see certain components, what their function is, what's the function of a ribosome. I hope everyone knows the function of a ribosome now is to make protein. We see an 80S ribosome in eukaryotes, a 70S ribosome in prokaryotes. What else? The ER is the factory of the cell. The Golgi is the membrane-bound organelle that routes different products to their end location, and then the lysosome is a sac filled with digestive enzymes. So go through everything. Chloroplasts we only see in plant cells. Plant cells have a cell wall. Animal cells do not have a cell wall. We have sterols. Just go through everything, and if you guys have questions, you know where to reach me.